Okay, well, hello and welcome everyone to our program on water issues in Nebraska. My name is Peggy Apthorpe, and I am currently working as an AmeriCorps with Conservation Nebraska. Conservation Nebraska is a statewide nonprofit that is dedicated to keeping our natural resources healthy. And one of the main ways we do that is through offering educational programs like the one that we are having tonight. This is the second in a series of 4 programs related to the water that we are co-sponsoring with the Nebraska Water Center. So we're very grateful to them for lending us their time and expertise to help us get the word out about water issues. Before I introduce our speaker, I want to remind you that your microphone is muted and your camera is off, so you can put questions and comments in the chat or the Q&A, and we will address them at the end of the program. Also, a short survey will pop up at the end of the program. We'd really appreciate it if you take the time to complete that, because it helps us with funding and planning issues. So it's my pleasure to introduce our speaker, Dr. Daniel Snow. Dr. Snow is the director of the University of Nebraska Water Sciences Laboratory, where he has worked since 1990. He oversees the Water Sciences Laboratory staff and operations using analytic and chemistry to help understand how water becomes contaminated and what we can do to prevent it. His work involves creating analytical methods to identify new or emerging environmental contaminants, and he is a real leader in the whole issue of environment and the water. So we're really pleased that he has agreed to spend time with us tonight, and I'll turn it over to him.
Well, thank you, Peggy. I really appreciate the chance to talk about water quality. It's a topic that's kind of near and dear to my heart. So I hope what I'm going to cover tonight will be of interest to you all, and maybe spark some more conversations about water quality and what we should do to protect our water resources in Nebraska. All right, hopefully everyone can see my slides. So just kind of a quick overview. I'm going to talk about Nebraska groundwater, and specifically about drinking water quality. I'm going to spend a little bit talking about a new class of insecticides called neonicotinoids from a surface water study that we did at the end of 2019. Talk about some contamination that's occurring, has occurred at the Alta-N bioenergy facility off site from that facility. A little bit about the Nebraska Groundwater Monitoring Network, and then finally a few slides about our Know Your Well program. Well, imagine, because you're viewing this presentation tonight, that you've no doubt seen some of the news articles about nitrate contamination. I'd say about half of my slides tonight are about nitrate contamination and about how our groundwater became kind of contaminated with nitrate and some other contaminants. So first, I want to talk about water quality. That's actually a subject that I teach at the university, and some of these slides are from the class that I teach. Water quality is really a measure of the suitability of water for a particular use. And we don't really think about that, but it's based on physical, chemical, and biological characteristics. And we make measurements, which is what I do for a living. I make lots of measurements as part of the research that we do at the Water Sciences Laboratory. And then if we're looking, say, at drinking water, we're comparing those measurements to guidelines for what is acceptable for an intended use. And this is a photograph I like to show to my students when we think about water quality. A photograph of Smith Falls in Cherry County, Nebraska. If any of you have ever been there, you've no doubt gone and seen the falls, and maybe even stood underneath the falls and found it to be quite exhilarating. I remember it was very cold. This is a spring-fed stream and probably very high quality, but we're not really sure until we make some measurements of that water if it's suitable for drinking. Well, many of you have heard of the Safe Drinking Water Act. I like to cover this in my classes, mainly because it really is what helps us to define what safe drinking water is. There was a survey back in 1969 of U.S. public drinking water systems, and that survey found about 60%, only about 60% met minimum health standards for drinking water. And this was conducted by the American Public Health Association. Really close to half of our public water systems did not meet minimum health safety guidelines for drinking water at that time. And about the same time, the Environmental Protection Agency was created. And then shortly thereafter, there was a study that found 36 synthetic chemicals, contaminants like chlorinated solvents in treated municipal drinking water from the Mississippi River for New Orleans municipal water supply. And they found that many of those contaminants ended up in people's blood. They were measured using the same tools that we use at the Water Sciences Laboratory to detect and quantify contaminants. And really that sequence of events led to a passage of the Safe Drinking Water Act in 1974, which allowed EPA to regulate the quality of public drinking water supplies. So currently there are about 90 different contaminants that are regulated under the Safe Drinking Water Act. Includes 16 inorganic contaminants such as nitrate and nitrite, 55 organic contaminants such as pesticides and solvents. There's 4 radioactive contaminants, 7 microbiological pathogens such as E. coli, bacteria, as well as 7 disinfectants and disinfectant byproducts. And what we're trying to do when we measure the concentrations of these contaminants is evaluate potential health effects arriving from long-term chronic low-level exposure. So we're getting down to, for many of these contaminants, to our ability to measure them accurately and consistently for low-level exposure toxicity. I think it's encouraging, and I want to point out that most public water supplies in the United States have a very low probability of exceeding a maximum contaminant level violation. What that means is that most of our public water systems provide very safe drinking water. In fact, the United States has one of the safest public water systems in the world. So let's talk about a maximum contaminant level. Here's an example for nitrate. That maximum contaminant level is set low enough to protect infants from methemoglobinemia or blue baby syndrome. They found that there is a possible association between chronic nitrate exposure during pregnancy and human malformations. There's also some evidence for teratogenic effects attributed to nitrate or nitrite ingestion, as well as some associations between nitrate and nitrite exposure, but really the data on the research is conflicting on whether nitrate actually or nitrite can cause cancer in humans. So really, we're sort of we're at a better position now than we were when the Safe Drinking Water was enacted, but we don't really know about all of the long-term consequences of low-level exposure to nitrate, nitrite, and other associated contaminants. Well, I show this slide really to remind everyone that our water is connected, whether it be surface water from a stream such as Smith Falls, as well as groundwater, which is what most Nebraskans use for drinking water. That groundwater in Nebraska is especially vulnerable to contamination from what we do at the surface, especially from our agriculture. And I'm going to go into that in a little bit of detail in the next few slides. But really, all of our water is connected to each other. It was shortly after I came to Nebraska and started my graduate program and worked at the Water Sciences Laboratory that the government actually officially acknowledged through passage of a law that groundwater and surface water is connected. And it's really true in this state, partly because of the geology underlying most of our soils. Well, I'm going to show quite a few slides from the U.S. Geological Survey. It's an agency or survey that shows that has done a lot of work on water quality for the United States. And one of the works showed the potential for nitrate contamination of groundwater. The map on the left shows the predicted nitrate concentration, red at 10 parts per million or 10 milligrams per liter at that MCL. And just recently recharged that's very close to the surface. And the map on the right shows a predicted nitrate for deeper groundwater used for drinking water, typically used by public water systems. And I think you can see in Nebraska, we've got a lot of red. We have a lot of areas in the state that have a high potential for contamination, both in the shallow, recently recharged groundwater and also for the deeper groundwater that we might ordinarily think is protected from surface contamination. Well, this is that same map, and next to it is a map of nitrogen fertilizer use in the last 115 years. And I think you'll agree that there is some similarity in the pattern of nitrogen fertilizer use and the areas of groundwater that are contaminated with nitrate. If I blow that last map up from 2015, you can see that the areas with the highest amount of nitrogen fertilizer that's been applied in Nebraska correlates very closely with what we find in the groundwater throughout the state with high nitrate concentration. So I think that really is strong evidence for really fertilizer contributing to nitrate contamination in Nebraska and the rest of the United States. Dr. Roy Spalding, who was my PhD advisor, and Mary Exner published a work looking at eastern Nebraska and the potential for contamination of local groundwater with nitrate. And what they found is that there was a much higher risk for groundwater nitrate in areas with a shallow water table, thin vadose and sandy soils with a high density of irrigation water or irrigation wells, as well as aerobic or oxygenated groundwater. And that's what these maps are showing, is a trend in increasing extent and higher concentrations of nitrate in eastern Nebraska. One area is in northeast Nebraska in Holt County. What's part of that includes the Brazil groundwater management area, as well as area along the Central Platte Valley through Grand Island. A study I was involved with, very recently published, looked at the entire state and correlated the high nitrate concentrations with a few other factors, including the density of irrigation wells and irrigation methods, as well as correlation with poorly drained or well-drained soils and depth to the water table. And what we found is basically the same conclusion using data from a variety of sources, that there is clearly a higher risk for groundwater nitrate contamination in areas with a shallow water table, a thin vadose, sandy soils, and high density of irrigation. So it really is agriculturally intensive land uses that lead to over our sandy soils and shallow water tables that has led to nitrate contamination. So sandy soils, high irrigation well density, and shallow water table, thin vadose are all the most important factors that lead to contamination in our state. So this just shows a sort of a snapshot of data from the Nebraska agrochemical contaminant database for Nebraska groundwater, showing the trend in concentrations for 3 counties in northeast Nebraska, around 15,000 samples from Antelope, Knox, Holt, and Pierce counties collected between 1970 and 2015. And what we don't see is what happened in the preceding 15 years or so, but there was a rapid increase in nitrogen fertilizer use that likely led to the high nitrogen concentrations in the shallow groundwater from this area. There was a publication that came out in 1979, actually, that documented the increase in fertilizer use in Holt County, one of the 4 counties that are shown in this graph. What this shows us overall is that nitrate came up very quickly, probably the early to mid 1970s, and then it just stayed high and gradually increased with every succeeding year. I'd say probably 2/3 of the samples on here are higher than that 10 milligram per liter nitrate maximum contaminant level. And the dotted line actually shows in these particular samples that there's a slight increase in nitrate contamination. All of that is consistent with the maps that I just showed. So we had the important aquifers and topographic regions of Nebraska. The Central Platte is the topographic region with the crosshatched valleys. And that corresponds quite nicely with the high nitrate in the groundwater throughout the Platte, Central Platte Valley. Using some maps from the 2022 Nebraska Groundwater Quality Monitoring Report, we can see that maybe not quite so bright of a pattern of nitrate contamination in these wells. These were collected between 2000 and 2019, 2,000 domestic and public water wells, basically showing the same trend that was illustrated in those earlier studies. Remember, these are just public and domestic water wells. But if we look at the irrigation wells, we see a very bright pattern of nitrate contamination, 2 times the maximum contaminant level or more from the irrigation wells. And it's because irrigation wells, we have a much larger number of samples, and they end up extending, the screens are extended throughout the aquifer typically. So they end up drawing on groundwater from regions or from areas in the aquifers that are fairly heavily contaminated. Well, it's not just nitrate. We also have a few violations for some geogenic contaminants such as arsenic, selenium, and uranium. The number of violations have gone down over time, but these are still persistently showing up in many of our public water systems. There are several public water systems in Nebraska that have had to install expensive treatment systems for uranium and arsenic. Some have had to drill new wells looking for lower concentrations of these naturally occurring contaminants that we believe are also being mobilized by our high intensity of irrigation and increasing nitrogen fertilizer use. Well, I sort of talked a lot about the public water systems because they're regulated under the Safe Drinking Water Act. But one thing that we know less about is domestic well water quality. Private wells are not regulated under the Safe Drinking Water Act. That is, they're not tested regularly, and there's no legal mechanism to require testing on a regular basis. So it's really up to the well owner to test their drinking water supply in rural areas. And what I wanted to point out in this figure is that those domestic wells are the ones that are typically screened closer to the water table in comparison to public supply wells. So if public supply wells are contaminated, it's much more likely that the domestic wells in the same area are also contaminated, mostly because they're more highly vulnerable than a public supply well would be. Well, it turns out there's a lot of people using private wells for drinking water across the United States. This map from the USGS shows the density of domestic wells use per person, people using domestic supply wells per square kilometer. In the eastern part of the United States, there's a high density of people that use private wells. We have a lower density in Nebraska, but because we draw very heavily on groundwater throughout the state, pretty much everyone that has a domestic well in the state their well is vulnerable to contamination from the surface. We have much less information about domestic well or private well water quality. One study that was done by the U.S. Geological Survey in the early 2000s looked at the quality of water from domestic wells in the U.S. And I think you can see the blue dots here, which cover the High Plains Aquifer System or Ogallala Aquifer System. It includes Nebraska. So they had quite a few samples from this region, and we can see what they found from that private well sampling. Roughly a quarter of the wells sampled contained one or more contaminants higher than that maximum contaminant level or the health benchmark safe level for drinking water. Inorganic contaminants, including nitrate, tend to occur more frequently than the organic contaminants, and that nitrate levels are greater than human health benchmarks in 4.4% of all of the wells in the United States that were sampled. E. coli and total coliforms were detected in 8 and 34% of the wells tested, and roughly 48% of the wells contained at least one contaminant outside the range recommended for aesthetic quality, such as very hard water, staining, say from iron or manganese, and unpleasant odor or taste. In general, many contaminants tend to co-occur, with the highest co-occurring contaminants being nitrate with diethylatrazine. Diethylatrazine is a byproduct of atrazine, a commonly used herbicide. And then they also found that nitrate was correlated with radon detection in domestic wells. This is a summary of the top 8 inorganic contaminants. As I mentioned, radon was one of the more commonly detected contaminants, followed by strontium. Arsenic was a very common contaminant in domestic well water. Manganese, of course, nitrate, uranium, boron, and fluoride tended to be those that kind of rose to the surface of the contaminants in domestic wells that would be exceeding human health benchmark and likely would be good for further study. Well, this was published, I guess, about 5 years ago, looking at the probability of arsenic that's above the 10 microgram per liter safe drinking water maximum contaminant level in domestic wells. And we can see that in the western U.S., especially in Nevada, as well as in some from Iowa and Texas, there's a greater than 0.5 probability of arsenic occurring in a domestic well. In Nebraska, we have some areas that are red and orange, as well as quite a bit that's yellow. And it's true, we have had some public water systems that are impacted by arsenic contamination, and it's highly likely that we have domestic wells that are also impacted by arsenic contamination. Well, we started measuring groundwater contaminants for a wider variety of contaminants through a Nebraska Groundwater Monitoring Network program in 2021. Altogether, we plan to analyze over 600 samples collected by Nebraska Natural Resources districts. Measurements will have included measuring nitrate, as well as fluoride and some other anions, arsenic, selenium, manganese, iron, boron, strontium, and uranium. And those contaminants were the same ones that I mentioned in the USGS study, as well as herbicides such as atrazine and atrazine degradation products, as well as some new classes of pesticides such as neonicotinoid insecticides and strobilurin fungicides. All of this data will go into that Nebraska agrochemical contaminant clearinghouse that I've been using, that is typically used for mapping groundwater quality across the state. So a little bit about the preliminary results from the first year of the sampling. Around 220, or about a third of the samples, were collected by 12 Nebraska Natural Resources districts. Average nitrate was around 8 milligrams per liter. We had quite a few samples that were well above 10 MCL. Average sulfate was around 100, and chloride around 20. Fluoride, bromide, and other anions were generally at the safe level, less than 1 milligram per liter. Moving on to those geogenic or naturally occurring contaminants like arsenic. We found arsenic The average arsenic concentration was fairly low, around 4 parts per billion or microgram per liter. Maximum was 22, about twice the human health benchmark or maximum contaminant level. Boron was another element that we were looking at. All the boron concentrations were well below that human health benchmark. Iron is pretty common in domestic wells, and we found iron in one sample that was almost 10 times that benchmark of 300 parts per billion. Manganese is also a pretty common contaminant in Nebraska groundwater. 50 micrograms per liter is that human health benchmark, and the highest concentration was over 1,400 micrograms per liter. Selenium is maximum concentration that we measured in the first year was about 70 micrograms per liter compared to that 50 micrograms per liter benchmark. Strontium, 4,000 is that benchmark, and we had a few samples that exceeded that benchmark level. And then there's uranium, 30 micrograms per liter benchmark, and the maximum that we measured was about 116 micrograms per liter. So there were some samples that exceeded the Safe Drinking Water Act human health benchmarks for these contaminants. Herbicides, a little bit fewer detections at high levels. We detected one sample of atrazine, which was above that benchmark level, and one sample of for metolachlor. Actually, metolachlor was at fairly high concentrations, but it was below that human health benchmark. So with respect to herbicides, we're not seeing as much detections as we are the other contaminants. We are, however, seeing traces of atrazine and the atrazine degradation products. And then the newer classes of pesticides, such as those neonicotinoid insecticides, just getting a feel for the concentrations that we might expect to find. Most of these were non-detects, but we did find some degradation products, as well as one of the insecticides. Looks like it was clethianidin at about 1.5 micrograms per liter. That was well below the health benchmark level of 200 parts per billion. So now that I'm talking about insecticides and fungicides, I wanted to spend a few slides talking about pesticide seed treatment. There are likely over 40 to 50 chemical treatments that are classed as fungicides, insecticides, nematicides, and plant growth regulators that are used as pesticide seed treatment. These include the neonicotinoid insecticides, as well as some strobilurin and azole fungicides that are found on treated seed corn. Clethianidin, that was the one that we detected in one of the samples from the Nebraska Groundwater Monitoring Program. We did an estimate of the amount of active ingredient per acre. So they're really using a very small quantity of any of these pesticides when they plant treated seed, less than or about 22 milligrams per acre as treated seed. So it's a very small quantity when you're using these compounds appropriately. Unfortunately, we are seeing basically an explosion in the use of clethianidin and other neonicotinoid insecticides. This map is from the USGS. They're monitoring geographically, they're monitoring the use of many pesticides, and this shows the density of the amount of clethianidin used in the United States in 2014. The bar graph shows that there's a great increase up until 2014. We actually likely have had an increase since 2014, but they're not tracking it as closely because treated seed is much more difficult to track for the amount that's used as if it was used, say, in a spray or a granular form. Well, we became interested in measuring these insecticides, the neonicotinoid insecticides, primarily because of their effect to honeybees. And this is a publication that was done by a graduate student in the entomology department at UNL, looking at thiamethoxam and methanomoxam in soybeans and how they might affect bee colonies up at the NREC, the Eastern Nebraska Research and Extension Center. So we found pretty significant levels of these neonicotinoids insecticides, and they were being transferred to honeybees that were kept on site. In a more recent study, working with another graduate student in biological systems engineering, we measured these same insecticides as well as some fungicides in runoff into several eastern Nebraska lakes and reservoirs, including Pawnee Lake, Holmes Lake, and I believe the other is, I can't remember the third lake. But anyway, the graph on the right shows the concentrations that were measured on average in parts per trillion, not parts per billion, but parts per trillion, as well as the chronic invertebrate limit for each of those compounds, clethoanidin, imidacloprid, and thiamethoxam. Overall, the concentrations were below what would be harmful to invertebrates, such as crayfish and snails in the streams. But there were occasions when the concentrations would exceed that invertebrate toxicity level. And then another student from biological systems engineering did a review article on these neonicotinoid insecticides and was interested in some of the degradation products from the clethoanidin and imidacloprid. And it turns out that many of these degradation products are even more toxic and probably more concerned than the parent compounds. For example, imidacloprid degrades into imidacloprid desnitro in the environment. And the desnitro form of the parent compound can have a greater mammalian toxicity, meaning it could be more toxic to aquatic life that are exposed to those degradation products. So finally, getting to even more news, there was a release of wastewater from the Alten bioenergy facility that contained fairly high concentrations of these neonicotinoid insecticides, as well as some fungicides that are used in treated seed corn. There was a rupture of a valve that caused release of around 4 million gallons of wastewater. And much of this flowed onto the Eastern Nebraska Research and Extension Center, photographs by Dr. Judy Roosmart. And then that followed with heavy rains that led to additional flooding. Nebraska Department of Environment and Energy had collected some samples and had those tested and found very, very high concentrations of several azole fungicides, as well as abamectin in this wastewater release. Our lab tested some of the grab samples from this wastewater release on the NREC site and found very high concentrations, especially of the degradation products, such as the imidacloprid desnitro, as well as thiamethoxam urea. So it ends up being a mixture of many compounds, including these degradation products that are likely to be of a concern from a toxicity standpoint. So as a result of that study, the University of Nebraska Med Center was given some funding to do some environmental monitoring. And we've been testing these monitoring samples, both grab samples and passive samplers from the watershed that had received the wastewater is disposed on cropland, as well as some wet cake that was applied as a soil conditioner. What these graphs show is that as we go from upstream to downstream through the Johnson Creek Reservoir, that we see an increase in the concentration of these neonicotinoid insecticides, consistent with more of a point source contamination due to wet cake application. Enric Creek is an intermittent stream, so we didn't get as many samples from that stream where that had received the wastewater, but it had by far and above the highest concentrations of the insecticides and the degradation products. So we've continued that monitoring that began in 2021. We had approximately 9 months of monitoring with grab samples and passive sampling. We also did some deep beta zone coring at selected wet cake and west wastewater application areas. And we're working with the U.S. Geological Survey and EPA to conduct additional sampling and explore ecological environmental exposure potential effects. We've been using these passive samplers as a photograph of these passive samplers where we can deploy it in a stream and it will accumulate contaminants over time. It gives us much greater sensitivity than grab samples would. It also can even out some of the variability that you might see from stormwater runoff. We have sampling stations up and down Johnson Creek and an unnamed tributary to help characterize how runoff from this land, from land application of waste products will affect surface water quality, and use both sets of data to help understand the release and dissipation of the contamination from cropland. And we now have almost 2 years of monitoring data, and what the first 2 years have shown us is that the concentrations are decreasing over time, which is a good thing. Results suggest that the pesticide loading from the wet cake and wastewater application is declining over time. As I mentioned, the USGS is doing some additional sampling. Their drinking water and wastewater infrastructure integrated science team is measuring the occurrence of these compounds, as well as a bunch of other contaminants in domestic wells from the Alton area. We have another USGS team that is sampling and evaluating impacts to aquatic organisms in the area streams. We hope that the results of this additional testing will be available in a year. And then I mentioned we wanted to do some deep coring. We have had 3 deep cores that were collected from the Eastern Nebraska Research and Extension Center. The graph in the middle shows the concentrations of clethoanidin in the unsaturated zone from the surface down to the water table. And we can see that the treated vadose had higher concentrations than the control. We're going to compare these levels in the vadose zone and try to predict how quickly they might be moving through the vadose zone to the water table. And we're interested in identifying any additional locations where wet cake and wastewater were land applied to investigate pesticide transport and degradation transport to the local groundwater. So I just want to wrap up with kind of a high note. This is a program that I've been involved with for the past 6 years. Know Your Well is a high school community science program. In the first 2 years or 3 years of the project, we had 150 students from 19 schools across Nebraska that collected and tested domestic well samples. They also sent the Water Sciences Laboratory split samples that they could use for comparison of their results. Our long-term plan is to develop a curriculum for teachers and then transfer leadership of the Know Your Well program to Nebraska Natural Resources districts. It's a pretty sophisticated citizen science program. Students screen samples using test probes and visual test kits and portable colorimeters. They measure things like pH, temperature, and conductivity, as well as coliform and E. coli bacteria, of course, nitrate, several metals. And in the first years of the project, we were able to do a strip method for atrazine. The split samples are tested, and then we provide a well analysis, a well owner analysis of all of the tests that were run at the Water Sciences Laboratory with those health benchmarks, basically to give them an idea of the quality of their domestic well. Students and teachers can access data that's stored online on a secure database on the Know Your Well website. And we keep owner identity confidential in all of this data. And then we provide training videos and promotional materials on the website, basically to grow the program. So here's an example of the laboratory results from 2017 to 2019. As I mentioned, we had almost 200 wells. I think it was 200 wells. And these are divided by school, showing a comparability in the graph on the right between the test kit as well as the laboratory method for different schools that participated in the project. And then on the right-hand side, we've got the arsenic concentrations that were measured at the Water Sciences Laboratory in comparison to the EPA maximum contaminant level. And right away, I can see that we have some wells that were testing above that 10 parts per billion maximum contaminant level. So we were very fortunate working with the Papio, Missouri Natural Resources District and several natural resources districts across Nebraska. And we have received funding for an additional 3 years of this project from the Nebraska Environmental Trust, Department of Environment and Energy, with some support from the Nebraska Water Center and Dougherty Water for Food Global Institute. We're working with a group from University of Nebraska Kearney Communications Department to do the promotion and network communications side of things and develop a new website and cross-platform application for data upload and retrieval. Eventually, expansion of this program will permit participation from more than 50 schools in Nebraska and testing over 1,000 domestic wells, hopefully within the next 2.5 years. And with that, I'll be happy to take any questions.
Thank you very much for that interesting program. I'm going to start with the ones that are in the Q&A. What 12 NRDs are participating in the groundwater monitoring project?
In the first year, let's see, it was, we got samples from, I can't remember off the top of my head, but this is, that was a 3-year program. So we had 12 NRDs out of all 23 NRDs that started in the first year. So eventually we'll get samples from all of the NRDs, and I don't remember off the top of my head with which 12 participated the first year.
Okay. Okay, is there a filtering system such as a reverse osmosis that will remove nitrates and other hazards from my well water?
Yeah, that's a great question. Typically, reverse osmosis is the one that most private well owners use. It's a good idea to couple reverse osmosis with a softener because hard water will affect the performance of a reverse osmosis system. So you need to soften the water first and then go through RO. Distillation is another purification method that works very well. It also has to be softened prior to running through a distillation line. So either RO or distillation are both good treatment methods for nitrate.
Okay. Is there any impact of irrigation using groundwater with high nitrate on the quality or quantity of crops?
That's also a good question. Nitrate is a nutrient. It's what causes eutrophication in lakes. So the crops actually use the nitrate in irrigation water the same as they would use nitrate from nitrogen fertilizer.
Okay. Why are you testing for copper? Don't water pipes impact the sample results?
Yeah, that's a good question. The reason we're testing for copper is there is a maximum contaminant level for copper. And we're interested in just like public water systems, we would be interested in copper contamination of drinking water from domestic wells that may come about because of corrosion of copper pipes.
With Nebraska not regulating FPAs in water, are you concerned about FPAs or PFAs? I'm sorry. Would you support regulation of PFAs? Have you considered adding testing of PFAs?
PFAs. Yes. PFAS. PFAS, I think it's
okay, yeah.
per and polyfluorinated alkyl substances. I think that's what that individual is asking about. And we do have a method. We actually put out a publication last November on a method for PFAS in wastewater. And we're currently developing a new method. It's based on EPA 1633 for PFAS and hope to be testing a lot more samples for this emerging contaminant.
Okay, so we have a few questions here. Do the NRDs also monitor the water quality of the public municipal water systems?
Public water systems? Is that the question?
Yeah, do they monitor public municipal water systems?
Mm. They don't directly monitor, but they do keep track of public water systems within their boundaries. Nebraska Department of Health is the one that's responsible to track water quality from public water systems, and then NRDs receive reports from the Department of Health if there are any issues with a public water system water quality.
Okay. And this is from Sharon. I love scientists. You are experts and I respect your detailed knowledge of these complex topics, but would it be possible for you to clearly demarcate with lines on your charts when water quality or chemical levels are okay and when they are not? The numbers are interesting, but not useful to mere mortals like most people.
Yeah, I could have added more MCL lines or health benchmark lines than I did on some of those graphs.
Hello, I'm curious to know if any of the water samples were additionally screened for viruses. UNO's virology lab course was rooted in screening Nebraska water samples for viruses.
We did not test any viruses.
Okay, another question from Megan. Also, suggestions for garden fertilizers, insecticides that are more safe for our water systems. Thank you.
The question is groundwater safe for irrigating gardens or food crops? Is that the question?
It's can you suggest garden fertilizers or insecticides that are safer for our water system?
Oh, okay. So where there would be a lower potential for contaminating. Okay.
Yeah.
Hmm.
Garden fertilizers or insecticides.
Yeah. Well, yeah, this is true for either homegrown vegetables, fruits and vegetables, or row crop agriculture. If you use too much of anything, it has the potential for contaminating water. So if you use a different product, if you're using more of it, that could lead to contamination. So really the take-home message is to use exactly the right amount that's required to control or fertilize your crop, especially for the soil, with respect to the soil type. So there are some products that are probably less likely to leach and contaminate water systems, but what it seems to happen more often is that too much of nutrients and pesticides are used, maybe under the wrong soil types and crop conditions.
Okay. Sharon says that she loves the program for high school students to do water testing. It's brilliant. Miles says, Dr. Snow, are you aware of any organizations doing remediation with artificial wetlands and mushroom compost to remove nitrates?
You know, I had a student from Lincoln Public High 2 years ago that grew some mushrooms, and she wanted to have she conducted some field experiments to see if it would remove nitrate from runoff water in the city limits. And it turned out that the mushrooms don't work that well for nitrate, but they work pretty well for removing phosphorus.
Interesting. When are the other webinars scheduled? I did put that in the chat, but the webinar on water quality, flooding, and drought in Nebraska will be on March 14th with Dave Aiken. And on April 11th, we have one on water and climate change with Rizal Mahmood. So you can also check out the Conservation Nebraska website at neconserve.org or follow us on Facebook, but we'll be advertising those very soon. Okay, how about testing for antibiotics since they are used in CAFOs?
Yeah, CAFOs, concentrated animal feeding operations. We actually have done a lot of work on antibiotics in livestock production systems. Antibiotics are used in livestock production and released through wastewater and manure. And we have quite a few research papers on that topic, which I would be happy to share. Anybody, if you have questions after this is over, you're welcome to email me and I'll try to answer questions and send you some of the papers on those topics.
Okay, let me just. I want to make sure I'm getting all of these.
I see a question if anyone's testing for lead. That's a great question.
Okay.
Lead, we don't test for in the domestic well sampling project because lead is very insoluble. There are conditions that would cause lead to be removed due to corrosion in plumbing. And I think that was the situation that was in the widely known in Flint, Michigan situation with their public water system. Is the water chemistry caused the lead that was already in the plumbing to become mobilized and contaminate drinking water in that system. But public or private water supplies are probably not highly vulnerable to lead contamination. And that's one reason we're not testing for lead.
Okay. Let me get my computer here to work. Okay, Harry is asking, I'm on the west side of Lincoln on 3 acres, and when first built and moved in 30 years ago, my nitrate tested at 8 or 9. We didn't do anything at that time, but it kept climbing, and about 10 years ago put in an RO, which dropped it to 0 to 1 or so. Without the RO, nitrates are 20 or so. However, it's only on our separate tap in our kitchen sink for cooking and drinking. We still drink. I'm losing his. Sorry about that. We still drink the water, well water, apparently from our bathroom to take medicine and a cup of water, minimal amounts overall. But should we avoid doing that?
That 10 milligram per liter maximum contaminant level is mainly to protect infants and pregnant women. So if you don't fall into that category, it's probably safe enough, even at 20 milligrams per liter, especially if you're not consuming very much of it.
Okay, how often should one test private wells?
Well, it depends. I guess I'm on a rural water system, and so our rural water is tested at least once a year. I would say private wells once a year is probably not too often.
Is there any regulation about fertilizer use in Nebraska, like quantity?
Yes. So natural resources districts have been working to educate producers, especially in areas where there's a high vulnerability for nitrate contamination. And I think we're slowly moving in a direction where producers are using less fertilizer and at the right time. So it's education that is hopefully leading to the point where we'll start to see a reduction in nitrate contamination in some of these areas that have seen high nitrate for a long time.
Does using high nitrate water on a home garden affect any of the crops?
Oh, as I mentioned, nitrate is a nutrient, so nitrate in your well water would actually be good for the plants.
Okay, let me make sure we got. Well, Miles says, wow, thank you. I think we got all of the questions. And I did put the information about the upcoming programs in the chat box, but we really want to thank you, Dr. Snow. This was a real informative program. Oh, okay. My coworker's telling me there's some at the top of the chat. So let's see here. Okay, well, I think, are you talking about our local municipal water? Lincoln Water sends water quality reports to users.
Not easy to read.
Not easy to read. What should we look for as red flags on these reports? Some of
Well,
the are
yeah,
national. Yeah.
they should print out the results and compare directly with their maximum contaminant level. So that's that EPA safe drinking water regulatory level. And if anything is getting close to or exceeds an MCL, that means that the municipality is not meeting the minimum standards for public water systems.
And I think this is the last question. Has a study been done to follow the clearance of nitrates or other contaminants a given well field following retirement or of irrigation? I recall irrigation was stopped in an area to provide additional water for the Republican River.
Yeah, I'm trying to think how that might work. I'm not aware of any study. I'm just trying to imagine what would be the consequences of not pumping. It seems like it should reduce if you stop pumping in a region, that you would over time see a reduction in nitrate contamination of the local groundwater. So that's a great question. And I think now I'm curious and I want to go look on the agrochemical contaminant database and see if we can find some places where that has occurred.
Okay, I think I missed a couple here. So anhydrous ammonia is only supposed to be applied in the spring, but many apply in fall because it is easier. Should that be enforced?
Yeah, so I think the question is, do we want to stop doing anhydrous ammonia application in the fall? My answer is yes. I think that it really is not a good use of fertilizer to apply it in the fall. As long as the soil is wet enough to hold the ammonia and clay rich, then it's fine. But I think what happens is with climate change, we've had warmer winters, and if we don't get the crops in, we can lose a lot of that nitrogen before it is used by the crop. So I really am not in favor of fall fertilizer application of any kind. And I grew up on a farm. I understand why they do it, but for water quality, it's really not a good idea.
Okay, how much chemical runoff do buffer zones collect from surface water?
Yeah, so grassed buffers are often used as conservation management practices, and they can remove nitrate that runs off in surface runoff from a field. They can also reduce the amount of pesticide concentrations that occur in surface runoff. I don't think they do a great job at removing nitrate that might be directed into the local groundwater.
What should be done to try to lessen nitrogen fertilizer use by farmers and other sources like CAFAs?
I'm not sure I understand the question.
I want to try to clarify. I know you have to do it in the chat, but you want to try to clarify it here? We'll go on. I'll go on to the next question. Do you know the effects of these early issues and the effect of thyroid issues in people?
Yeah, I don't actually, but I was reading an article on the effect of drinking water contaminants on thyroid function and forms of thyroid cancer the other day. I don't remember what I was reading, but I know that there is a connection between drinking water quality and thyroid. I can't recall it off the top of my head.
Yeah. Have mandatory buffer zones been discussed for Nebraska?
Not to my knowledge. I teach a water quality strategy class, and 1 year that was proposed as a way to improve water quality, was to basically use a CRP program and try to make sure that the CRP areas were buffers, grassed buffers, to reduce losses from storm runoff.
How close are some of our rivers getting to be in like Iowa?
That's a good question. I grew up in Iowa. I think the geology of Iowa is quite different than Nebraska. And consequently, most of the nitrate occurs in runoff. Not all of it, but most of it occurs in runoff from fields. Now, there are going to be some public water systems and some private wells that are affected by surface runoff. And of course, there'll be some areas that will be affected by nitrate contamination in the local groundwater. How close are some of our rivers getting to be like that? I think overall nitrate is fairly low, especially in the Platte River, because our water comes from the Rocky Mountains, the Colorado and Montana Rocky Mountains. So when it enters the state, the nitrate concentration is pretty low. I think once it gets to the other side, here in eastern Nebraska, we end up seeing higher concentrations, just because of all the agriculture that water goes through.
This is another question related to the thyroid. I was born in New Orleans in 1971, and I have had thyroid function issues since I was a kid. Has there been any traction of diseases in humans and the increase of cancers in children?
I've seen more studies on this connection between drinking water quality and human cancers, including thyroid function. So, I think we have more data that shows an association between contaminants in drinking water and human health effects, like thyroid function. I don't know when do we get enough data to make a clear connection? I think you have to pair those studies with toxicology studies that are done in a laboratory, say with animals, to really demonstrate that there's a cause and effect from the exposure.
Okay, this is Harry is trying to explain, I think, his question, but he was wondering about concentrated feedlots, whether we should have more regulations for them. Let me see, I've got to go up.
Yeah, I think I understand the question. Yeah, animal feeding operations are regulated. So any runoff from a feedlot has to be directed into a stormwater retention structure, and that water has to be contained. It can't be discharged into a stream. Where we might need more regulation is in monitoring manure that's applied to cropland and making sure that we're not over fertilizing areas that are already receiving plenty of nitrogen and phosphorus from animal manure.
Okay, and I think this is the last question. Are you aware that the water runoff is causing dead zones in the Gulf of Mexico?
Yeah, actually, I did my master's in Louisiana back in the 80s, and we already had a dead zone in the Gulf of Mexico at that time. So that's been pretty well known for a long time.
Hey, I think, Dr. Snow, we really appreciate your taking the time to do this, and you did a wonderful job. If people do have more questions and they want to contact you, do you want to give us your email or?
Yeah. You want me to put it in the chat?
Yeah, that would be good. And then also for the upcoming water programs that Conservation Nebraska and Nebraska Water Center are doing, they will be up on our Facebook page or on our website very soon. But the first one will be March 14th. Again, water quality, flooding, and drought in Nebraska by Dave Aiken. Thank you everyone for attending. I think we had almost 60 people here. Oh, yes. Okay. And then we have the little feedback survey. If you can take a few minutes to do that. And I want to thank my Zoom host, Sarah McLean. She's one of my fellow AmeriCorps who always acts as the tech person when I do programs at any rate, because it's all kind of new to me.
Yeah, thank you all for joining. And then, yeah, thank you, Dr. Snow, for the presentation. That was super informative. And, water is so important to our state. It's good to get the word out there about what we can do to help.
Right. Thank you. Okay. If your email's up there, people can go ahead and email you with additional questions. It was really an informative program. Thank you.
You're welcome.
For taking the time on an evening.
Yep.
Okay.
Bye.
Bye, folks. Have a good night.
Yeah.